So let us start another theory class, Autacoids. First we have to go through the definition of Autacoids, and thereafter we are going to discuss about the histamine and its analog. So first we have to define the Autacoids. The word Autacoids, the term Autacoids derived from two Greek words, auto. So we all know this is auto. What is the meaning of auto? Auto means self. And the another Greek word, echoes, means drug, a remedy, or a medicinal agent. That means that this autoquats is self-remedy. So regarding function is concerned, autoquats are performing various physiological as well as pathological functions. Regarding drawback with the autoquats is concerned, they are having very short half-life. So this is another feature that have autoquats have. This autoquats is also called local hormone, and usually we are going to compare it with the endocrine. What the local hormones means? Local hormones means it have the paracrine effect. What it means? It means कि these autoquats are acting nearby where the autoquats are released, or it work at vicinity where it is secreting. So we have to summarize these all points that we already discussed and define that autoquats are the substance produced by various tissues and have the paracrine effect. So that is why it called local hormone. Its action is rapidly but briefly and have several physiological and pathological functions, and importantly they also serve as a transmitter, a modulator, and also it mediate the or it modulate the function of neurotransmitters are called autoquats. So this is all about the autoquats. Now we have to go through the classification of autoquats. Regarding classification of autoquats is concerned, the autoquats are classified based upon the chemical structure. So upon the chemical structure, autoquats are divided into three groups on the basis of its chemical nature. If they are protein in nature, it's called peptide autoquats. If derived from lipid, it is called lipid derivative autoquats. And if it is an decarboxylated amine, it's called decarboxylated amine autoquats. So there are three types of autoquats, namely biogenic amine and also called decarboxylated amine autoquat. If you go through the name, then you will find it is the removing a process of removing of uh, carboxyl group from an amino acid, and therefore as a resultant we receive the autoquats are called decarboxylated amine autoquats, and that is also called biogenic amines. The example under this autoquads are, or the autoquads under this class are histamine and 5-hydroxytryptamine. 5-hydroxytryptamine is also known as serotonin. So this is all about the example under the decarboxylated amine autoquads. Now we have to go through those autoquads that are protein in nature. So that is why they are called peptide autoquads. They are called polypeptide autoquads. And these are kinins, angiotensins, tachykinins, and cytokines. So these are the example of protein autoquad, also called peptide autoquads, also called polypeptide autoquads. And amongst that, the angiotensin is very, very important, like angiotensin 1, angiotensin 2. Why? Because these angiotensin are participating in blood pressure of the individual or the patient. Another important under this peptide autoquads are cytokines, which include interferon, interleukin, and growth factor. Under the kinase, the example are bradykinin and calidin. This bradykinin as well as calidin is also very much participating in regulation of the blood pressure. So this is also very, very important kinins. And the last one under these examples are tachykinins. The examples are substance P and neurokinin A. So these all about the peptide autoquads. Now we have to go through the lipid derived autoquads. So name is clear cut indicating they are derived from the fatty acid or lipid. Therefore they are called phospholipid or lipid derived autoquads. And now we have to go through the example. The important lipid derived autoquads are, the first one is the eicosanoids. So eicosa means 20. So in this about 20 fatty acids are present, and or you can say that derive from the 20 fatty acid. So that is why they are called eicosanoids. Under these, the autoquads are prostaglandin, thromboxane, and leukotrienes. So these are the leukotrienes itself is telling this the having the three double bond. Then the last example under the lipid derived autoquads is platelet activating factor. So this is all about the classification of the autoquads. Now we have to start with the first decarboxylated autoquads. Under this, the example is histamine. So we are going to discuss about the histamine, its analog, or you can say the agonist. So we are in this class we are going to discuss about the histamine, its agonist, and let us start with the word histamine. The word histamine is derived from the term or name histamine is derived from a Greek word histos, and histos is itself means the tissue. So it is derived from the tissue. It is a biogenic amine, and what are the function or mediator that a biogenic amine autoquads histamine have? The histamine is participating in inflammation, in anaphylaxis, as well as a very good important physiological function histamine which participated functioning is gastric acid secretion. So this is another very, very interesting example, interesting physiological function of this histamine. So how these histamine are acting? Histamine are acting by attaching with their receptor. So about four important receptor of the histamine are there. So that means when this histamine is going to attach with the receptor present on the cell, and thereafter they are going to produce the physiological function after the conformational changes produced by the receptor, either its activation or inhibition. So this is about the receptor. That means histamine is going to attach with the mainly four type of receptor: the histamine 1, that is H1; histamine 2, that is the H2; histamine 3, that means H3, and so on up to the 4. Now we have to go through the function of the particularly the H1 antagonist. So as we know, what is the meaning of antagonist? Antagonists are the substance which are going to block the function of other endogenous substance. So that endogenous substance is not going to attach to their respective receptor to produce its physiological function. So antagonists are the substance which are going to block the function of the endogenous substance to attach to their respective receptor to produce a physiological function, and that is why they are going to call the antagonist. So therefore, what are the physiological function that the histamine produce, whether through H1, H2, H3, H4 receptor? If we are going to use their respective antagonist, and their physiological function get inhibited. The example are H1 receptor. So H1 receptor, when histamine is going to attach with the H1 receptor, so therefore the histamine is going to produce the physiological function like allergy, then anaphylaxis reaction. Then this is also going to sometimes produce nausea, motion sickness, insomnia, then sometimes the symptom of asthma. So if these all physiological function of the histamine is going to produce through the H1 receptor, so these physiological functions should be inhibited when we are going to use its antagonists, so that are called competitive antagonists. So this is about the H1. Take another example, H2 receptor antagonist. H2, when histamine is going to act on its receptor H2, and therefore this H2 receptor is going to produce the or H2 receptor is participating to produce the hydrochloric acid from the parietal cell. So what will happen when there is blocking of this H2 receptor by its antagonist? So the acid should not be produced, so therefore it is used as a antacid also. H2 receptor antagonist used as a antacid. Now we have to go through the discovery, or you can say the history of the histamine. So when we have to go through the history of histamine, the first important scientist, which part here I am going to mention one very important point, is that before its biological action of histamine has been recognized or disclosed, or biological significance was recognized, like histamine, it was first prepared. So before its isolation, or before its biological significance was known, it has been first prepared in the laboratory, and thereafter it has been isolated, or its biological significance was recognized. And if we have to go through that history, then regarding this is concerned, the histamine is first prepared synthetically in the year 1907, whereas it has been isolated from extract of the ergot in 1910. So it is very much clear. First, the histamine was synthesized. However, it has been isolated later on. So regarding this endogenous substance or autocoids is concerned, I mean histamine autocoids is concerned, histamine was first reported or first discovered in the year 1910 by Henry Dale, Sir Henry Hallett Dale. So Sir Henry Hallett Dale has recognized this, first discovered this in the year 1910, and this was isolated from the contaminant of our gut generated by bacterial action, and that was the histamine. Histamine, so it was first discovered. Later on, in the year 1927, the scientist Best and its colleagues also isolated histamine from fresh tissue of liver as well as the lungs, and therefore he named it on the basis of its isolated, from where it has been isolated, from the tissue. Which type of tissue? Liver and lungs. So it, the tissue, the Greek word for the tissue was the histo. So therefore the name of the histamine was coined by the Best and colleagues in the year 1927, and the name was the histose based from its isolation. Again, later on in the same year, in the year 1927, Lewis and its colleague also explore more about the histamine, and he told that the histamine is also liberated from the skin that was injured skin, and also histamine was released when there is antigen antibody reaction, that is the anaphylaxis hypersensitive type 1 reaction, that is the AIgE mediated. And recently it is well known, known fact about the histamine is that histamine is also modulating the function of neurotransmitter in the CNS and PNS, peripheral nervous system. So this is all about the history, and the last one point under the history is the lead law and Dale and lead law. Its detailed pharmacological effect of the histamine was given in the year 1953 by the scientist Dale and Laidlaw, and he told about its activity on the smooth muscle and the blood vessels, particularly vasopressor action. So this is all about the uh, history. Now we have to go through the chemistry of the histamine. So regarding chemistry of histamine is concerned, chemically histamine is possessing one imidazole ring and one amino group. So these amino group and the imidazole both are connected together by two methylene group. So chemically, if you want to explain what is histamine, histamine is possessing one amino group and one imidazole ring, and both are connected by two methylene group, is called histamine. What other thing the histamine chemically have? They are monocation N gamma H tautomers. So this is another important point. Other than this important, another point is that the histamine is main having more affinity towards the receptor like H3 and H4 receptor than the H1 and H2 receptor. So this is very important thing is that histamine is possessing more affection towards the receptor like H3 and H4 as compared to the H1 and H2 receptor. So this is very important. And now we have to go through the agonist. Agonist means having the similar physiological function like histamine. So now we have to go through the agonist of histamine for H1. So we call it H1 receptor agonist. Then for the H2 we called H2 receptor agonist. Then H3 and H4 we called is the H3 H4 receptors agonist. Now we have to go through the agonist of the H1 receptor. So if you are recalling the name of histamine, then this become very easy to recall its agonist. So name of the H1 receptor agonist you have to put before histamine 2-methyl and add it and tell 2-methyl histamine is a H2 receptor antagonist, and when you remove this 2 from this 2-methyl histamine and add 4, so the name is 4-methyl histamine. So 4-methyl histamine is agonist to the both H3 and H4 receptor. So this is very very important. Other example of H1 receptor agonist are 2-pyridyl ethylamine, 2-thiazolil ethylamine. Regarding the agonist for the H2 receptor is concerned, the best example is dimaprid. Other are amphetamine, then impromidine, then H3, H4 receptor agonist, the example are alpha methyl histamine, imitate. So this is all about the agonist that we have discussed. Here I am going to mention two very very important point is that earlier initial and initial stage, dimaprid and 4-methyl histamine was earlier more known as 2H receptor, H2 receptor agonist. So dimaprid and the 4-methyl histamine are considered as H2 receptor agonist, but later on it was found that this dimaprid and methyl 4-methyl histamine, dimaprid and 4-methyl histamine are possessing about 100-fold more affinity towards the H4 receptor than the H2 receptor. So this is very very interesting and important. Now we have to go through the distribution and biosynthesis of the histamine. So here interesting thing about the biosynthesis of the histamine is concerned. The histamine, first we have to tell about the distribution. It distributed well not only in the mammalian tissue, it is also found in the venom, it found in the bacteria, it found in the plant. So you can say the histamine is widely distributed, and therefore that is almost all mammalian tissue have the histamine. So this is very very important. If you go through the where the high concentration of histamine is present within the body, then in the cerebrospinal fluid the amount is significant. In the body fluid like plasma and other body fluid, it is generally very low. But when you have to consider the mast cell, the amount is predominant amount of histamine is present in this mast cell, and therefore, where the mast cell is distributed, the probability of histamine release is more, and the place where the mast cell found are skin, in the bronchial mucosa, in the intestinal mucosa, near blood vessels, lymphatic vessel, within nerve, urinary tract. So this is all about the distribution. Regarding the distribution, other point of the distribution is that this is also distributed in the basophil in the blood vessel, mast cell in the tissue, then the within the platelets of some species, and other cells are nervous tissue. This is very very important. Also in the gastric mucosa. So this is very very important for the human being life. So now we have to go through the synthesis and the metabolism of the histamine. Regarding synthesis and metabolism of the histamine is concerned, you have to see here, when we have gone through the histamine, we found that so first we have to go through the synthesis of histamine. So since histamine is decarboxylated, that means ki carboxyl group is removed, and that one is synthesized from one amino acid histidine. So histamine was synthesized from an amino acid histidine by removing a carboxyl group, and therefore as a resultant the compound is formed, which we are calling is histamine. So histamine get formed. So this is about the histamine synthesis. That histamine is going to synthesize from an amino acid histidine by removing a carboxyl group, that is called decarboxylation. So decarboxylation is occurring. So the name of the enzyme is decarboxylase, and because the decarboxylation of the histidine occur, so this is the histidine decarboxylase. So this occur in the presence of histidine decarboxylase, and therefore histamine was synthesized. Now this is about the synthesis process. Now we have to go through the metabolism. So there are two pathway of metabolism of the histamine. The pathway one, that is predominant pathway. In this predominant pathway one, there is mainly the methylation occur, and in the phase 2 metabolism of the histamine, the deamination occur, and also the final phase ribose get attached. So let us see the first one, that is the predominant phase of the metabolism of the histamine. So histamine is going to first added with the methyl group. So methyl group is get added. Therefore, the name of the enzyme is the transfer of methyl. So the name is the N-methyl transferase, and therefore the name of the component is, as a resultant form, is the N-methyl histamine. So N-methyl histamine is the first component formed during the metabolism of histamine in the presence of methyl transferase, N-methyl transferase. Thereafter, in the final step, in which there are synthesis of methyl imidazole acetic acid, N-methyl imidazole acetic acid, and this was occur in presence of amine. We are going to call it monoamine oxidase B. So N-methyl histamine get converted into N-methyl imidazole acetic acid in presence of an enzyme MAOB, that is the monoamino oxidase B, and therefore it get excreted out through the body. Now the second metabolite of this histamine is through deamination. So in this deamination, it is removing the amino group. So this amino group is removed, and therefore the reaction is called, the pathway is called deamination pathway. In this deamination pathway, and this is occurring through the oxidation. So oxidative deamination pathway, this pathway is not occurring much more. In this, histamine is first converted into the imidazole acetic acid. This imidazole acetic acid is synthesized when histamine get deaminated by an enzyme deamine oxidase, and therefore amino group get removed, and a resultant compound is imidazole acetic acid. And finally, in this imidazole acetic acid, there is addition of ribose sugar, and therefore the name of the resultant compound is imidazole acetic acid riboside. So this is the final product formed from the next second phase of the or second pathway of the histamine metabolism. So therefore this is two type, ring methylation is occurring, and second one is the oxidative deamination occur, that we already discussed about this in detail. Cheap source of histamine synthesis as well as storage. So where histamine is synthesized more and as well as it get stored is the cell is called mast cell. This is not the most cell, this is the mast cell. So in mast so this is okay, this is correct. In most of the most tissue is mast cell, which is synthesizing as well as the storing in the secretory granules of the mast cell, as well as in the blood. Basophil is also synthesizing and storing it in the secretory granules. So there are these are the other than this non-mast cell are which are storing the histamine or forming the histamine are gastric mucosa, neurons in the CNS, then the epidermis. So this is all about the metabolism and synthesis pathway of the histamine. Now we have to go through how the histamine is going to function and how it is going to be released. So this we are going to discuss, very important. So see here, as I think we all have gone through the anaphylaxis or type 1 reaction. So what will happen in type 1 reaction? In type 1 reaction, the mast cell is, this is the mast cell, and in this mast cell antibody is attached. So when this antibody like IgE is attached and that is sensitized by the histamine, that is sensitized by anything. So because this mast cell is having histamine stored in the granules, so this is the mast cell. So what will happen when the respective or concerned antigen is going to attach with the antibody IgE present over the H1 H1 present on the mast cell, then what will happen after the reaction of the antigen and antibody, they get burst, and therefore this is going to release the histamine, and therefore accordingly the released histamine is going to produce the function that we are going to call as the anaphylaxis, also going to produce the allergic reaction. So this is about the one function. Other function that the histamine and as well as its release, that we are going to discuss one by one. But among that one very important function is that histamine is also act on the smooth muscle, that is vascular smooth muscle is also, bronchial smooth muscle is also, and also it and therefore it may produce the allergic response. So histamine is participating in the type 1 hypersensitivity reaction through mast cell, and also it is going to act on the smooth muscle, maybe the bronchial smooth muscle, maybe the gut smooth muscle, or the smooth muscle of the blood vessel, the vascular smooth muscle. As well as one most important point with the histamine function is releases the hydrochloric acid. How this is going to release the hydrochloric acid? So this is a mast cell. So no, this is the parietal cell. This is the parietal cell, and the parietal cell having the H2 receptor. And if over this H2 receptor this histamine is going to attach. So when histamine is going to attach with the H2 receptor present on the histamine is going to attach to the H2 receptor present on the parietal cell. Therefore, what will this will do through activation of this H2 receptor, they are going to give the increased the protein GS. This is this protein is a G protein coupled receptor protein. So once this GS is going to produce, this is going to finally produces the cyclic AMP level. So this cyclic AMP level is going to raised, and this cyclic AMP is going to act on the proton pump. So here is the proton pump, and this cyclic AMP is going to activate the proton pump, and through proton pump the hydrochloric acid is releasing. So this is the pharmacological action of the histamine over the parietal cell. So on the parietal cell, histamine is going to attach with the H2 receptor. Therefore, it's attached to the guanyl GS protein, and therefore it activates further an enzyme adenyl cyclase, and therefore adenyl cyclase is going to convert the ATP into cyclic AMP, and therefore it finally having the positive effect on the proton pump inhibitor, and that proton pump inhibitor is going to release the hydrochloric acid. So this is the physiological function of the histamine over the H2 receptor that is participating in the SCL secretion, and also the histamine is modulating the neurotransmitter release. Now we have to go through the pharmacological effect of the histamine. In this we are going to see how this histamine is acting at molecular level. So you have to listen it, and you have to understand whether whether you are understanding or not. That is very very important. So uh, all the histamine receptor like H1 to H4 means H1 receptor, H2 receptor, H3 receptor, H4 receptor. So these all receptor of the histamine are GPCR, G protein couple receptor. So these all are G protein couple receptor. So when histamine is going to act indirectly on the blood vessel, so this is the blood vessel. So on the blood vessel, when the histamine is going to act indirectly, then it is going to attach with the endothelial cell, that is the H1 receptor of the endothelial cell. So indirectly or indirect action of histamine is attachment of histamine to H1 receptor present on the endogenous receptor, and as a resultant they activated the GPCR protein, that is the GQG11 GQ protein, or GQG11 protein, and upon activation of GQG11 this is going to give rise to pathway phospholipase 3, IP3 and calcium pathway. So what is in nutshell in this endogenous histamine acting on the endothelial cell of the blood vessel is that in nutshell acting through the GQ oblique 11 protein, they are going to release the inositol triphosphate IP3, and therefore it raises the intracellular calcium of the the intracellular calcium level of endothelial cell of the blood vessel, and therefore finally it helps to release the nitric oxide through the enos, endothelial nitric oxide level. So this calcium is going to helps to release the nitric oxide by activating the nitric oxide synthase in the nitric oxide synthase enzyme, and therefore the release of nitric oxide occur. And this nitric oxide is not nitric oxide is going to act on other another cell of the vascular smooth muscle. We call it the vascular smooth muscle, that we all discussing about the indirect effect. How the first histamine is going to act on the H1 receptor through GQ protein to release the intracellular calcium, and raised amount of intracellular calcium is participating in releasing nitro oxide, and the released nitro oxide get diffuses, and therefore they are going to act on the smooth muscle, and therefore when this nitric oxide is going to act through the cyclic GMP and is going to produce the relaxation. So this is very, very important. So this is indirect effect. First on the endothelial cell to release nitric oxide, and after the release of nitric oxide, it is going to nitric oxide is going to act on the this nitric oxide is entering in the vascular smooth muscle, and this in nitric oxide is going to activate the guanylyl cyclase, another enzyme. Therefore, it increases the cyclic GMP, and now cyclic GMP is participating in the relaxation of the cyclic GMP. So this is very, very important. So how they are acting, that I have explained. And in contradictory, see here, if this nitric oxide is acting directly, so the action become different. How the action become different? So when the histamine is going to act again on the same H1 receptor, but this receptor, H1 receptor, should present on the smooth muscle, not on the endothelial cell. So when the histamine is going to act on the H1 receptor of the smooth muscle, similar to the earlier GQ pathway, they are going to increase the intracellular calcium level. There in endothelial cell, they are performing the relaxation of the blood vessel or dilatation of the blood vessel. Here the action is the direct one, rather it is releasing the nitric oxide. Here the raised calcium level is acting on the myosin protein to produce the contraction. So this is just contradictory type of the property of the histamine through the same H1 receptor. So that you have to understand. Now we have to go through the H2 receptor and H3 and H4 receptor. Regarding H2 receptor is concerned, regarding H3 and H4 receptor is concerned, when the histamine is acting on the H2 receptor present on the parietal cell, so what will happen? Or on the smooth muscle, then what will happen, that you have to see. So when histamine is going to attach with the H2 receptor on the parietal cell or on the smooth muscle, what will happen? It is finally going to increase the cyclic AMP level, that we have discussed in the gastric acid secretion through the proton pump. And so similarly in the smooth muscle, they are also going to act like that. So what will it do? H1 histamine is going to act on the H2 receptor. It activates the GS protein, and after activation of the GS protein, it go through a pathway that we are calling it the adenyl cyclic AMP protein kinase pathway. So activation of H2 receptor on the H2 receptor on the smooth muscle is going to relax the is going to relax the smooth muscle. How? Not contract. How this is going to relax? They are going to act on the G activation of GS protein, and this GS protein is going to activate one enzyme which is called adenyl cyclase, and this adenyl cyclase is going to convert ATP into cyclic AMP, and this cyclic AMP get accumulated and raised in the intracellular, and therefore this is going to act through the protein kinase A, and this protein kinase A or cyclic AMP is having the negative effect in contraction, therefore it produces the relaxation. So this is very, very important. Now we have to go through the H3 and H4 receptor activity. So when the histamine is going to act on the H3 or H4 receptor, their activity is quite opposite to the H2 receptor activity. So here the protein get activated is inhibitory. So the name of the protein is GI slash or oblique O, that inhibit the opposite action. So it inhibit the adenyl cyclase, and therefore it lowering the cyclic AMP. So this is about the H3 and H4 receptor. Now we have to go through the more detail pharmacological action of histamine through the H1 and H2 receptor. So first we have to go through where this is present. So H1 and H2 receptor is present in the periphery as well as the in CNS, and it have widespread effect on the smooth muscle and the gland, and also have and that we already discussed, the local hormone is having the local effect. So autocoids are local hormone. Likewise the histamine is also in this you can say the local hormone. So it can produce itching, as well as upon stimulation it will going to increase the secretion of nasal mucosa. Now we have to go through the activity of histamine through H1 receptor on the various type of the smooth muscle. The but main smooth muscle is the smooth muscle of the blood vessel. So before that we have to go through the activity of the this on the bronchial and gut smooth muscle, bronchial smooth muscle, gut smooth muscle, and when this is going to act through this H1 and H2 receptor, they are going to contract it because it have direct action. So what will happen if the bronchial smooth muscle or the gut smooth muscle is this? So this one is either having H1 receptor or H2 receptor, this H1 and H2 receptor. So what this is going to do through H1 and H2 receptor, this is going to produce the contraction. How this one is going to produce the contraction? This H1 receptor through H1 receptor, this histamine is going to attach with the H1 receptor, and when this is going to attach with the H1 receptor, this is going to go through the GQ receptor, GQ protein, and therefore it is going to raise the calcium level, intracellular calcium level. So when the intracellular calcium level get raised, this is going to produce the contraction of the muscle. So when it produces activity directly through the G1 receptor, so it acts through the GQ protein. So it acts through the GQ protein, and it give rise to raised intracellular calcium level and contraction occur. And whenever it acting on the H2 receptor present on the smooth muscle of anyone, then again this is going to contract it. How this is going to contract is regarding the through this vascular smooth muscle. So this H2 is going to produce in case of H2. This contract the smooth may, and this is so sorry, this is mainly through the not through the H2 receptor. This is only through the H1 receptor. This is only through the H1 receptor, not through the H2 receptor. Now we have to go through the its relaxation over the blood vessel, that we a small blood vessel. It's the direct activity that we are not going to discuss. Now we have to go through its gastric acid secretion through the H2 receptor. So here, when the histamine is acting on the H1 receptor, present on the bronchial smooth muscle, present on the gut smooth muscle, they are going to contract it through the GQ calcium pathway. So we are calling it the this pathway GQ phospholipase C IP3 calcium pathway. As a resultant, calcium raised, and therefore it is going to produce the contraction of the bronchial smooth muscle and the gut smooth muscle. And when this histamine is going to act on the parietal cell, the receptor is for the histamine, the receptor is H2 receptor. So therefore, when the histamine is going to attach with the H2 receptor, so what will happen, that we already discussed. So it activated the H2 receptor. Therefore, it gives the through the GS protein, because it is GPCR. So through GS protein, it activates the enzyme adenyl cyclase, and because of the activation of adenyl cyclase, cyclic AMP is formed, and this cyclic AMP is going to relax the smooth muscle. But this is about the parietal cell. So in the parietal cell, what will happen? In the parietal cell, the raised cyclic AMP level, so raised cyclic AMP level is going to act on the raised cyclic AMP is going to have the positive effect. This raised cyclic AMP level through this pathway is going to have the positive effect on proton pump, that is present here on the same parietal cell, and proton pump is going to release the hydrochloric acid. So again I am going to repeat the same thing, how this is going to secrete the HCl. So again the histamine is going to attach with the H2 receptor present on the parietal cell, activate the GS protein, and it stimulates the adenyl cyclase to raise the intracellular cyclic AMP level. This cyclic AMP level having the positive effect, plus effect on the proton pump. So on proton pump is present on the parietal cell, and that is participating in the HCl secretion. So therefore positive effect is it means ki it have positive effect on the HCl secretion. Now we have to go through the action of the histamine through the two receptor, H3 and H4 receptor. So H3 and H4 receptor is present in central nervous system. H3 receptor is promoting the sleeping. So when there is a blockage of H3 receptor by any antagonist, they produce the wakefulness. So this is very, very important, that when the histamine is going to act on the central nervous system through the H3 receptor, they are going to produce the sleepiness. And when you are going to use any blocking this pathway by antagonistic, so it is going to produce wakefulness. So this is very, very important. H4 receptor is primarily present in the cell of hemopoietic system like eosinophil, basophil, and various type. Here very, very interesting finding is that what is the effect of histamine on its own release through the histamine. See here, when histamine is acting on the H2 receptor, when the histamine is acting on the H2 receptor, and so what will happen? It also initiated H2 receptor. It also initiated the negative feedback in, and because of the negative feedback, this is going to send the message, don't release more histamine. So histamine is increasing the cyclic AMP level through H2 receptor, and this increased cyclic AMP level is having negative feedback on release of histamine. So when there is more cyclic AMP through this pathway H2 receptor, there is also inhibiting the synthesis and release of histamine, not synthesis, release of histamine. And the another one interesting is that the H3 and H4 receptor is that we already discussed. This is when the histamine is acting on the H3 and H4 receptor, so it is act like inhibitory protein GI/GO, and therefore it is going to lessen the synthesis of cyclic AMP. So it reduces the synthesis of cyclic AMP. So therefore it has somewhat opposite type of activity than the H2 receptor, because in H3 and H4 receptor they are telling decreasing the cyclic AMP level, and the decreased cyclic AMP level is stimulating the release of histamine. So these are the contrary activity. Now we have to go through the most important activity of or the function of the this histamine is its effect on the cardiovascular system. Effect of histamine on cardiovascular system, that we already discussed. In majority of blood vessel, particularly the resistant vessel, it dilate. It produce relaxation and dilate it. So therefore, because of dilatation of the majority of resistance vessel, this is going to lower the blood pressure. So histamine through both the H1 and H1 H2 receptor pathway is going to participate in the relaxation of the vascular smooth muscle, that dilate it and lowering the blood pressure. How? That we already discussed. Histamine is acting on the endothelial cell, and when the histamine is acting on the endothelial cell through H1 receptor, going to produce nitric oxide. This is indirect effect, and this nitric oxide is now again acting on the smooth muscle, vascular smooth muscle, and therefore through cyclic AMP level, rising the cyclic AMP level to no, 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 cyclic through not this nitric oxide, not through the cyclic AMP level, but rather through cyclic GMP pathway, that is relaxing the blood vessel. So nitric oxide is going to act through the cyclic GMP production more occur, and therefore it is going to produce the relaxation. So what happened through through H1? So here, see here, this nitric oxide is going to release, and this nitric oxide is going to act on the vascular smooth muscle, that we already discussed, and going to relax. How? The nitric oxide is activating the another enzyme, that we call it guanylyl cyclase, and this guanylyl cyclase is going to convert the GTP into GMP, this GMP, cyclic GMP, and this cyclic GMP is going to relax the vascular smooth muscle. This is this way. Now we have to go through the activity of the histamine through H2 receptor, the same, the relaxation. How? this histamine is going to attach with the H2 receptor. It activates the GS protein, and therefore it activates the enzyme adenyl cyclase. So it raises the cyclic AMP, and when cyclic AMP get raised through protein kinase pathway, they are going to produce the relaxation. So this way the histamine is going to relax the resistant blood vessel and produce the lowering of blood pressure. So this is very, very interesting and important. However, as we already discussed, if the histamine is going to act on the H1 receptor directly, it contract it. How this is going to contract it, that we already discussed. This is going to contract through again by the this activating the H1 receptor. This H1 receptor is going to activated. This is direct action. So this is going to activate the GQ protein, and therefore through this intracellular calcium level get increased, and therefore through myosin they are going to produce the contraction. So this is very, very important. Now we have to go through the most important property that we already this is important one, and that we are calling it the triple response of the histamine, and that was discovered by the scientist Lewis. So we are also calling it the triple response of Lewis. What is this? In this triple response of Lewis, what is going on? We are introducing the intradermal injection of histamine. So upon introduction of intradermal injection of histamine, they are going to produce the triple response, means three response. First is the red spot, second is the edema, second is the red flush, that is also called flare, and third one is the wheel. How this is producing triple response, that we are going to explain. When you are introducing histamine intradermally, as we are already discussed, histamine is going to dilate the blood vessel, and because of the dilatation of the blood vessel, there is some efflux of the fluid also occur. So when there is efflux of fluid occur, when vasodilation occur, so vasodilation means ki more blood is there, so more blood you have to see like a red spot will occur. And once its red spot occur, later on it flare through the nerve ending, and when it get flare, it proceeded, it give more area, then this is called a red flush, and finally it comes and it appear in the form of wheel. So we are going to call it the wheel. So there are triple response of the histamine after its intradermal introduction are red spot, formation of red spot at the site of injection. Then there it's taking more area, it is called flare up, that is red flush, and it form lastly the wheel, that we call it the wheel formation. There is no role of histamine as a clinical use, and they are mainly used as a diagnostic agent to see the hyperactivity of bronchial smooth muscle, as well as its a practical utility to keep the skin, allergic skin, as a positive control by giving the histamine. So this is all about the